before we start, I must show you this, which I don't know if anybody saw this on television Sunday night. So this is a show that CBS has been hyping for the last month or two. Came on right after the football games, which is the only reason I saw it. But this guy says, "My name is Watson. I'm a clinical geneticist."
I love it.
And then the first part of the plot was him explaining how autosomal dominant inheritance works. I wouldn't bet on this being a big commercial success. But anyway, I guess we've made it if we have a TV star that claims to be a clinical geneticist.
Morris Chestnut is a clinical geneticist.
Pardon?
I said Morris Chestnut is a clinical.
Yeah, I guess that's his name, right? Morris Chestnut. And
That's so
those of you that
funny.
have some interest in history will know that they stretch things a little bit because he apparently went to Europe to work with Sherlock Holmes, which, as best as I can tell, was sometime around 1880, just before Mendelssohn was rediscovered.
Oh, God.
Hopefully, at least the public will now understand clinical genetics.
Yeah, well, if they get it right. If they get it right. Okay, so I'm not going to, I gave you an article, or I mentioned briefly last time, a paper that looked at the increased gene discovery rate using long read sequencing. And this paper just came out. In fact, it's in press. It's not, I guess it's not in the hard copy yet. This is a much better paper than the one I gave you. So maybe I haven't decided yet, but we may use this for our journal club. And it comes from Broad. It's pretty well done, and the details are there, but we can talk more about it then on Friday. Next. Okay, some characteristics of Mendelian disease. So we're going to focus initially on just these three features, which are really important. First is that there's a lot of heterogeneity in Mendelian disease. And we think that's a single gene is pretty straightforward, but in fact, there's a lot of heterogeneity, and that heterogeneity contributes to two classic features of Mendelian disease. One is variable expressivity, which is individuals with the same Mendelian disease, in quotes, have it to different degrees of severity. In one family, it may present in infancy with desperately sick neonate, and another family, it may present in early adult life with not such a sick person. So that's called variable expressivity, and we'll talk more about that in a minute. And then there's this sort of mysterious feature of Mendelian disease called penetrance, which is, if you look at all the people that have the disease genotype, some of them have the disease and some of them don't. Okay? So the question is, why do the ones that have the genotype don't have the disease? Okay? We'll talk, we're beginning to understand why that is so. All right, so allelic heterogeneity, and these are two contributors to genetic heterogeneity. Allelic heterogeneity is when you have multiple mutations causing disease at a single gene locus. So, for example, for cystic fibrosis, we now know of over 2,000 disease-producing mutations in CFTR. Cystic fibrosis is a little unusual because about 60 or 70% of individuals have the same allele, that's delta F508. But in the other 30 or 40% of disease alleles, there's all different kinds of mutations, all of which cause loss of function. But because the different variants may have different effects, different magnitude of effects on the function of the protein product, you can see right away that might explain variable expressivity. This individual might have CF and have two alleles that are only modestly or not so severely affect
Recording in progress.
delta F508 function, and they're going to have a milder disease, whereas two individuals with complete nulls will have a sort of a much more severe disease. And one of the ways you can get at that, think about that, is if you notice that if you look at multiple affected individuals in the same family, and allelic heterogeneity is the source of the variable expressivity, what would you expect for those multiple affected individuals in that family? Lauren?
Sorry, what was the question again? Could you repeat it?
So if you're trying to get at variable expressivity, and you have a family with multiple affected individuals, and the degree of variable expressivity is due to allelic heterogeneity, what would you expect for those individuals in one family?
That they would have similar alleles.
Right, absolutely correct. So you can say that breeds true, right? So if you have a proband that's mildly affected, then you'd expect the next individual in that family would also be mildly affected. Now, if it's some other explanation for variable expressivity, that won't hold. So it's sometimes worthwhile to ask, in a given family, is there the degree of expression similar or disparate? Okay. All right, and then the second big source of genetic heterogeneity is locus heterogeneity. That's where you have what medicine says is the same disease, but it's caused by different mutations in different genes. So that's a locus heterogeneity. Okay? And then the interesting aspect of locus heterogeneity is it predicts different components of the same biological system. You can think that disease results from failure of a biological system, and then different genes contribute proteins in that same biological system. So loss of function of the system leads to the disease. Next slide. So here's an example of both. So this is a clinical syndrome called Robinow syndrome. They have hypertelorism, wide space eyes. They have skeletal dysplasia. They have hypoplastic genitalia. And both autosomal dominant and autosomal recessive forms have been described. It's one of those ones, if you go to OMIM and you ask how is this inherited, OMIM will say there's some forms that seem to be inherited as an autosomal recessive trait, and some forms that seem to be inherited as an autosomal dominant trait. Okay? So the folks at Baylor, led by Jim Lusky, collected a large series, 21 probands for this disorder, and showed that all of them had defects, or nearly all of them had defects in the Wnt receptor. The Wnt receptor is a complicated system, shown here 1, 2, 3, 4, 5, 6, 7, at least 7 genes as components of that system. And then they showed that functions in non-canonical Wnt signaling. And then they showed that different kids that had Robinow syndrome had mutations in the different components of this biological system. So that's, what kind of heterogeneity is that? Locus heterogeneity, right. So then the next thing you want to ask is, well, is the illness the same in all of them? Maybe the individuals that have mutations in DBL1 or DBL3 are milder than the folks that have mutations in some other component, Wnt5A or whatever. So you can go back clinically, and now with the ability to dissect the phenotype, you can ask to what extent is the phenotype similar for depending on which gene is responsible. And sometimes, you can imagine maybe the protein encoded by one of these genes works in another system as well, and that has Robinow syndrome plus, and so you can begin to dissect the clinical phenotype. The more you dissect it, the better understanding you have of your individual patient, what the prognosis is, how best to treat it, and so forth. And similarly, they also have allelic heterogeneity. You see that there are different mutations in different components of this system. Next slide.
How would you describe that gradient of phenotype presentation due to locus heterogeneity?
How would I describe it?
Or so like between severe and less severe, is there like more accurate?
Yeah. So if you're going to go after Robinow syndrome in some detail, what you would want to do is to set up some objective parameters of the phenotype, and then score those objective parameters, and then ask how does that scoring outcome compare to which mutation is responsible for the Ravenel syndrome in that individual. But I think you have to be objective in the way you score the phenotype. And they went on to say, okay, let's look at all the patients in which the Ravenel syndrome is due to mutations in ROR2, which is one of these, ROR2 is this gene, encodes this protein. And they found some aspects of the phenotype that were more problematic in ROR2 Ravenel syndrome. So you begin to sort of dissect things and get a more accurate understanding of the diagnosis in your particular patient. Laura?
So just to clarify, like with locus heterogeneity, within those you can have allelic heterogeneity.
Yeah,
Apple.
you can have both. Yep. In fact, this is a rare disorder, so you would expect that. You would certainly expect allelic heterogeneity.
Right.
And whether you expect locus heterogeneity depends on how complicated the system is. But most proteins don't operate in isolation, so they all work consistently. Eventually, I think, in the future, you guys will do this, we'll develop what I would call an alphabet of biological systems. I think I mentioned this before. So this would be a biological system. It's associated with a set of phenotypes and so forth. And eventually we'll know maybe 1,000 biological systems. And then we can begin to look at mutations in all those biological systems. Next slide. Okay, another place you can look at variable expressivity, or what's been studied very well, is in cystic fibrosis, and Gary Cutting and his colleagues have been instrumental in this. You know that CF, although we typically think of it as a lung disease, is actually a multi-system disorder. In addition to inflammation and decreased pulmonary function, you have abnormal sweat, you develop cirrhosis in the liver, you have exocrine dysfunction, more diabetes. Neonates are at risk for neonatal intestinal obstruction, and males are usually infertile. So it's a multi-system disease. Next slide. And allelic heterogeneity is extensive. So part of that variable expressivity and pleiotropic effects of the mutations in CFTR are due to allelic heterogeneity. Next slide. There also are remodifier genes, and so if you look in the column on the right, you can see what fraction of the variability seemed to be due to which particular mutations you had to CFTR, and the lighter purple are the difference is genetic modifiers. Now, if you're segregating a genetic modifier in a family that's also segregating CFTR, remember I said that if all the variability is due to allelic heterogeneity, you would expect it to breed true, right? What would you expect it if the family is also segregating a modifier, a variant and a modifier locus? Well, it's a little more complicated, right? Because let's say you're heterozygous at the modifier locus, you inherit the CF, and then it depends on which genotype you happen to inherit at the modifier locus. You got it? So it's more complicated and harder to figure out, but you can do it. The way Gary Cutting and his team did it, they took advantage of that most patients with CFTR all have the same mutation, delta F508. So they said, here's a collection. They actually collected 1,000 patients that had delta F508. And then they looked for phenotypic differences amongst those set of 1,000 patients. And they used that to do actually linkage studies and found locations for modifier genes and then traced them down. Okay. You could do that for, what's another disorder you could do that for?
Progeria?
Yeah, progeria. Almost all have the same mutation. What else? Sickle cell disease. And achondroplasia, right. I keep telling Julie to do that. Next time you see her, tell her to do that. Excellent. And then environmental variables is the third big source of variables. And certainly in CF, secondhand smoke is a big variable in the severity of disease. Next up. Okay, let's turn to penetrance now. Remember I said penetrance is when some fraction of people who have the disease genotype don't have the phenotype. And remember, penetrance is all or none. It's either penetrant or not. Okay? So you can see a pedigree like this. Next. Next. Oh, back. Oh, there's more step. But okay, so here's somebody who's heterozygote in the first generation and is affected. In the second generation, heterozygote as well, but unaffected. And then the third generation, heterozygote and affected. Okay? So they're segregating the same allele. They're all in the same pedigree, but that middle person is non-penetrant. Now, the first thing you would ask is, could this just be variable expressivity? If I look at that patient carefully in an informed way, can I actually see that they are affected just more mildly than their two relatives? But there are people that you look at them every way that you can imagine and you can't find evidence for disease, and yet we know they have the disease allele. Okay? So they're truly non-penetrant. So, and what I like to think about it is not only does it sort of tell you something about the biology, but it suggests a way you can treat it. Because whatever's going on in this person is a way to treat this disease, right? So understanding lack of penetrance or non-penetrance may give you insights into treatment. Can you imagine how this might happen? Well, interallelic complementation is a possibility. Maybe something about the second allele in a dominant disorder complements for the disease allele. Imprinting, maybe imprinting is involved. Maybe the allele, the expression of the allele depends on the parent of origin who transmitted it. There could be some sort of environmental trigger. What do I mean by that? Well, think the best example is the simplest one, G6PD deficiency. People with G6PD deficiencies do fine unless they eat fava beans. That's the environmental trigger, and then they hemolyze. Okay, so there may be something like that. Could be a two-locus model. Some other locus is influencing the development of the phenotype or something else. Now, next slide.
Do modifiers fall into that two locus?
Yeah.
Okay.
Yeah. So I'll show you. So here's what I would call a single locus model. This is worked out for what's called PBX6 associated congenital scoliosis. You can see these patients have very severe scoliosis. See the severe angulation in their spine. This gene was, this phenotype is shown to be due to loss of function mutations in TBX6. It's inherited as an autosomal dominant, but with a high degree of non-penetrance. Okay? So you would get a pedigree like this, shown here, and it turns out that TBX6, normal protein coding copy of TBX6, is on different haplotypes. Okay? And you can score the haplotypes by single nucleotide variants along the length of the gene. So here, there are certain TBX6 haplotypes that express TBX6 at a high level. Okay. Strong alleles, you might call them. And there's a less frequent haplotype that expresses TBX6 at a much lower level. It's normal qualitatively normal TBX6, it's just expressed at a lower level. This is sort of like thalassemia of the TBX6 gene. Okay? I call these wimpy haplotypes. Okay, so TCA is the wimpy haplotype. This family is segregating a deletion of TBX6, but that person has the deletion and doesn't have a phenotype. And on the other allele has a strong haplotype, CTA, marked by CTA. This person has the phenotype, inherited the deletion allele from the father, and inherited the wimpy allele from the mother. So it has some normal TBX6, but not enough, it gets the phenotype. And that person, Stiff, over here, also inherited the deletion allele from the father, but inherited the mother's strong allele, CTS6. So this person's not. Okay, so those are differences at the same locus that influence penetrance. Okay? And there are now several examples of this. And we know that although levels of expression were worked out very well at the hemoglobin locus and the severe phenotypes are called thalassemia, there are many other examples now where different haplotypes of a normal protein coding gene are expressed at different levels, and that may influence what goes on in Mendelian disease genes. And you can always score the haplotypes by looking for single nucleotide variants around the length of the. Next slide. Now this is a two-locus model for explaining penetrance. First is a one-locus model, this is a two-locus model. This is a paper from Rick Lipton's group, and they were looking at congenital cranial synostosis. So you know the skull is develops from plates of bone that are connected by connective tissue that can stretch so that the head grows and your sutures fuse in mid-childhood. If they fuse prematurely, then the head gets deformed because the brain has to grow. Okay? And the shape of the skull is determined by which suture fuses. So if the sagittal suture, this one, fuses, then the head grows front to back, but not side to side, and you get a long skull. And if the fuses the metopic suture here, or the coronal suture here, then the skull tends to grow laterally and not grows laterally. Okay. So they had a hypothesis that craniosynostosis was a Mendelian phenotype. Next. So they did a cohort of 132 trios plus 59 additional probands without parents. They did whole exome sequencing, and they found a number of variants in a gene called SMAD6. Actually, in this cohort, they found a total of 13 different alleles of SMAD6 that can also function. But what they noticed was that this showed 60% incomplete penetrance. So 60% of people that had the presumptive non-functional SMAD6 did not have cranial systems. Only 40% did. So they asked what was the explanation for that, and they noticed that there was a common variant in a different locus, different chromosome, BMP2. BMP2 was already known, bone morphogenic protein 2 is already known to influence sutural growth in the skull. So they looked at the two genotypes. So here are 14 people that have the risk allele at SMAD6, or the causative allele at SMAD6, and the risk allele at BMP2, and they all have craniosynostosis. Here are some people that have the causative allele at SMAD6, but they don't have the risk allele at BMP2. 3 of them have craniosynostosis, but 13 of them are non-penetrant. Okay? And then here, these individuals do not have the causative allele at SMAD6. They have the BMP2 risk allele, and none of them have craniosynostosis. Okay? So you have the SMAD6 causative allele, but it can only be expressed in the setting of someone who has the risk allele at BMP2. And the paper's full of pedigrees, and you can go see that and stuff like that. So that's a two-locus model for penetrance. So it's an interesting phenomenon. It helps us understand disease better. It suggests if we knew, for example, how BMP2 works, perhaps that would be a way to treat these patients, have to do it in utero, and diminish their craniosynostosis. Yes, Carter?
How much is this multi-locus model integrated into carrier testing and other things like that in the clinic? Is it known if you're a carrier for this?
Yeah, it's not easy, right? Why could they do it? They could do it, first of all, because they had whole exome. They weren't just genotyping a disease gene. Then they were thinking in a smart way about this, and it was already known that the protein product of BMP2 influenced sutural growth, so it was a good candidate modifier gene. Okay, so it's been worked out, but not in many circumstances. But you guys will have the tools. Next slide. Okay, so another cool thing about Mendelian disease is sometimes the mutation and the perturbation of the function of a particular gene suggests a way to treat certain diseases. So it's like Mother Nature trying to tell you something. You just have to be smart enough to understand what she says. So these are sort of naturally occurring loss of function mutations that predict the treatment of relevant effect and safety. So one is this gene HAO1, which encodes an enzyme in glycosylate metabolism, and when it's inhibited, it prevents the appearance of a pretty dire phenotype called primary hyperoxaluria. There's not very much known about that. It's not a major problem. A better example is this gene called PCSK9. PCSK9 was first studied because in large studies it was shown that gain-of-function mutations in PCSK9 increased plasma cholesterol. So it was that said this gene has something to do with cholesterol levels. Helen Hobbs and her colleague Jonathan Cohen then looked to ask, well, if gain-of-function mutations in PCSK9 cause increased cholesterol, are there loss-of-function alleles in PCSK9 that reduce cholesterol? And they looked at a large series, and they actually found that heterozygotes with loss-of-function variants in PCSK9 had about 50% normal cholesterol. Okay. So what does that say? Well, maybe if I develop a drug that inhibits PCSK9, I can treat hypercholesterolemia. Next slide. So that's been done in different ways. This is using an RNAi drug, and here's the normal cholesterol in the untreated people, and here's the cholesterol in the treated people, and they get about a 50% reduction in plasma cholesterol. This is independent of statin drugs. So this could be used additive to statin drugs. So all of that was because they followed up on this rare Mendelian phenotype. Next. So what are some challenges? First of all, what are the number of Mendelian disease genes? Understanding the way the phenotype results from the mutation in the gene. It's easy now to find the mutations in the gene. We have the phenotype, but sometimes it's very difficult to connect the molecular defect to the phenotype. That usually requires experiments. And we want to improve the solution rate. We've already talked some about problems using whole exome sequencing. We want to integrate newer sequencing technologies, and we already mentioned today that paper using Oxford Nanopore and OMP. We want to work out these oligogenic etiologies. We want to be clever about functional testing. We want to understand Mendelian disease genes in phenotypes like fetal loss and complex traits. And we want to continue to understand penetrance and modifier genes. Next slide. So let's just talk for a minute about the number of Mendelian disease genes. Now, I already told you that about almost exactly 5,000 Mendelian disease genes are known. So it's about 25% of the total number of protein-coded genes. Next slide. So by definition, these are genes in which some fraction of variance in this gene produces a highly penetrant phenotype. Next. How many phenotypes? Next. So OMEM currently lists about 8,500 phenotypes described, and there's about 200, 250 new phenotypes a year coming into OMEM, so the number of phenotypes keeps growing. We already mentioned some phenotypes are conditional on environmental variables. For example, there's a defect in a herpes simplex receptor, and if you have a certain allele in that herpes simplex receptor and you get exposed to herpes simplex, you get very severe encephalitis. If you don't get exposed to herpes simplex, as far as we know, there's no consequence at all for it. So it's a little bit hard to follow the segregation because everybody's not exposed to herpes simplex, fortunately. Encephalitis. G6PD deficiency, MK deficiency, lots of examples of this. And it turns out that we said how many phenotypes per gene, about a third of the currently known Mendelian genes explain more than one phenotype. Next slide. So if you make a plot like this is back when the number of disease genes were not as many as currently, but you just simply plot the number of disease genes versus the number of phenotypes for which those disease genes are associated with. Most are associated with one phenotype. So think of phenylketonuria, mutations that phenylalanine hydroxylase cause PKU, but we don't know any other phenotype associated with that gene. But look here, there's some genes. Look at this one. I'm this far out here. A small number of genes produce as many as 13 phenotypes. These are 13 disorders that if they walked into a genetics clinic, we'd say that patient has this, and that patient has that, and never really think to combine and say that they're different mutations of the same locus. So there's some really interesting biology going on in these genes that can cause multiple phenotypes. And it usually, I believe, happens when the protein that's encoded by these genes has multiple domains, multiple functional domains. And so where the mutation is and the nature of the mutation, loss of function, gain of function, and so forth, perturbs the function of the protein that requires that particular domain. Remember I showed you the slide of the two different alleles, two different kinds of mutations at FBN1. One set causes FBN1 loss of function and you get classical Marfan syndrome. But mutations in another, in a different part of that gene that affect a certain domain in fibrillin-1 cause stiff skin syndrome, which is almost the opposite of Marfan syndrome. So it says that there's a lot of interesting biology going on in this protein, and I understand it. Next slide. So now we could ask, okay, if you've got 25% of the protein coding genes that cause Mendelian phenotypes, what's your best guess at what fraction of the total protein coding genome could be associated with Mendelian phenotypes? Anybody want to make a guess? We've been at this now, trying to find these genes. Remember, the genomic method came along in 2009. So we found, since 2009, we found roughly 25%. Carter.
I mean, maybe they're really complicated systems and it's hard to suss out exactly what they do, but I think like Richard Dawkins would probably argue that every gene should cause some change in phenotype, otherwise we would have ejected it from the genome already.
Right. Not only Richard Dawkins, but David Valley would argue the exact same way. For exactly the same reason. You know, evolution, if the gene doesn't do anything, mutations happen at random, and eventually that gene will be inactivated and lost. Next. Okay, so some people, though, say, oh no, it's going to be a relatively small fraction of the total protein coding genome, maybe 25-30%. Next. So, and we're actually at roughly 25% right now. Now, the curve's still going up, so I arbitrarily flattened this out. The curve is still going up. At a slow rate, though. Next slide. So, Richard Dawkins and Dave Valley and Carter would suggest the curve's going to look more like this, right? Nearly all genes. And why? Next slide. Next slide. It's an evolutionary argument. And we would say also that phenotyping is key. You're not going to recognize a Mendelian phenotype unless you do a very informed kind of phenotyping. So one reason for not recognizing the Mendelian consequences is maybe we just don't measure that phenotype very well. Next slide. Now, what about mice? Remember I said the Knockout Mouse Project, we're knocking out every gene in the mouse and asking what are the phenotypic features, and they have a standard set of phenotyping parameters that they measure. And the bottom line is that almost every gene that's been knocked out in a mouse, if you look carefully, has a phenotype. So this is another vote in favor of the Dawkins-Valley hypothesis. Next slide. Next. Now there is, however, this interesting phenomenon. I just mentioned this for completeness sake. There's a study done in a highly inbred population in Pakistan, and with a sequencing of a large number of individuals, nearly 2,000 exomes, and 2,000 200,000 exomes and 20,000 genomes. And these are all, quotes, healthy individuals. Now, they weren't phenotyped carefully, but they were walking into the clinic to give their blood, okay? So that's the criteria of healthy. And the interesting thing was they identified a subset of genes, 1,815 genes, in which the individuals were homozygous. Remember I said it was a highly inbred population. Homozygous for a loss of functional allele, and yet they seem to be normal. So these folks said, well, this human knockout, this is sort of equivalent to the mouse knockout project, suggests that you can lose function, have homozygous loss of functional alleles at a certain loci and have no effect on the phenotype. That's an interesting observation. I don't know what the follow-up has been on this. The question I would raise, well, maybe you have to stress the system to see the phenotype. Maybe you have to look more carefully to see the phenotype. There's lots of explanations, but it's a very interesting population. And I meant to see if there's a paper by Karaszewski more recently than 2020, and I have not. So somebody can check that out. Maybe we've got some progress in this. I don't know. Next slide.
Couldn't that also be due to environment in the environment?
Yeah, it could be like you need an environmental trigger and they don't have it, or they escape it. So if the fraction of Mendelian, so we've argued that I've argued the fraction of Mendelian genes is large. So why are they difficult to identify? And actually, we're identifying more and more every year, but it's not going up as fast as you might guess. Next. So the answer everyone always says, I should have asked you first, is unrecognized developmental lethals. In other words, mutations in these genes cause early developmental lethals, so we don't even know that the phenotype has occurred. Now, I argue that that's not the case. There's no doubt that there are developmental lethals, but if you look at genes for which are known to produce developmental lethals, and you look at what we've learned, a good one is the Rett syndrome gene, MECP2. We find individuals that escape the uterine carnage and are born alive, and they have a phenotype. And the thing is, they have inherited alleles that are just more less severe. So you predict that there's allelic heterogeneity, and that the developmental lethals are more severe alleles, and people that have the milder alleles escape, get out of the uterus and are born alive. So I don't think, we have to look harder, but I don't think this is the answer. Next slide. We studied in the Mendel project, we studied a cohort in which they had spontaneous abortuses that had normal chromosomes. And so we said this might be a good population to find Mendelian causes of developmental lethality. They collected the families by collecting the abortus material and the mother. That was, I would argue, the way a non-geneticist would collect the families, because every time we found something in the abortus material that wasn't in the mother, we didn't know if the father had it and he was fine and he transmitted it, or it was de novo in the fetus and it was the developmental lethal. That study went nowhere. Have to design your experiments. Next slide. So incomplete or insensitive phenotyping, technological limitations. Next slide. Buffering, robustness, and redundancy of biological systems. What do I mean by that?
Overlap between different genes in terms of function.
Right, so the fact that we are here as open systems exposed to a lot of environmental variables, and we tolerate those for the most part, that's because we have a lot of homeostatic systems that control blood sugar, control oxygen, control all of these things. And those homeostatic systems are the products of our evolutionary history, and they're pretty robust. So they're built to maintain normal physiologic homeostasis and earlier normal development in the face of some sort of insult, be that insult environmental or genetic. Next slide. Next. So you should develop the mindset of developmental and homeostatic vulnerabilities apparent when the system is stressed. So one way to see these vulnerabilities that may not be apparent initially is to stress the system. Next slide. So remember this pedigree I showed you, the adult onset OTC deficiency. Next slide. So remember, this guy who came in at age 32, near death, was 32 years old, and he was thought to be a normal individual, and here he was with a severe mutation in OTC, not a complete loss of function. And actually it was a regulatory variant, so the little bit of OTC that he made was qualitatively normal, so it could function. And his homeostatic systems were able to maintain normal levels of ammonia for the first 32 years of his life until he got this bad infection, remember, and his doctor treated him with steroids as well. So he got a real push on his homeostatic capacity, and it failed. And all of a sudden, we see he has a Mendelian phenotype. So that's our buffering and homeostatic capacities. Next slide. So one way to find more Mendelian phenotypes and Mendelian disease genes is to develop controlled ways to stress the system and bring out variation in the biological systems. Next slide. Now, even so, the relatively slow rate of Mendelian disease gene discovery suggests we might be missing some large class of causation, right? We've had a big, rapid increase in the number of Mendelian disease genes, and it continues, but it's not going like many hundreds of Mendelian disease genes a year. It's like 150, something like that. So one big set of genes, and I put this in particularly for Kirby, are the link RNAs. So you know that there are more than 10,000 link RNA genes in the genome. And less than 5 have been associated with Mendelian phenotypes. And even amongst those, the evidence is not super great. Of course, we don't sequence the link genes. We don't look by whole exome sequence. We don't look at them, number one. And number two, for many link RNAs, we don't know what they do. So even if we sequence them and found variation, that variation would be very hard to interpret in most cases, and therefore hard to correlate with a Mendelian phenotype. And this references a review for a modern review for link RNAs, pretty good. Next slide. And we know that the link RNAs are transcribed by Pol2. The transcripts are polyadenylated, and the current state of the art shows that transcription of certain link RNAs, maybe a large fraction of them, I don't really know what fraction, can perturb the transcription of close by other genes, let's say protein coding genes. And it turns out that the cis-acting regulatory elements that regulate the expression of the link genes can also influence the expression of nearby protein coding genes. So this paper was just came out, I think it was in December in the New England Journal, and they described 3 patients that had similar phenotypes. They had microcephaly, they were severe developmental delay, they had cortical atrophy, so see here this is an MRI, this is the brain, all of this is fluid around the brain, that's because the cortex is not what it should be. And they had a facies that was a long philtrum, turned up nose, so forth. So they looked like a recognizable syndrome, and they did a whole genome sequencing, and they discovered, next slide, that all 3 of those patients had deletions, different deletions. This is an expansion of up here, but all 3 deletions essentially took out this long link RNA gene that's called, for some reason, Chasser. I don't know why. But, and the Chasser link RNA is right next to a protein coding gene called CHD2. CHD2 already had a Mendelian phenotype associated with it, so it was a known Mendelian gene. And the phenotypes that were known for CHD2 were loss of function phenotypes. Mutations in CHD2 that cause loss of function. So they argue that perhaps these deletions cause overexpression of CHASR2, of the CHD2. Next slide. So remember we said you could find variants along the length of the gene and find the haplotype. So you could look at both the expression of both alleles. And this is a variety of cells and tissues from 2 patients. And the dark blue column is the allele, the CHD2 allele that has the adjacent deletion of CHASR. And the light blue is the transcript from the CHD2 allele that doesn't have the adjacent CHASR deletion. Okay. So what you can see is that those alleles that have the deletion of CHASR2 have increased expression of CHD2. And found the same thing looking at protein quantification of CHD2. So they argue that this Mendelian phenotype is due to overexpression of CHD2 due to a mutation, a loss of function mutation in the adjacent, very closely adjacent link RNA CHASR. I think that's very cool. Next slide. So they conclude that CHASR encodes a conserved link RNA. I didn't show this data. Deletion of CHASR results in a distinct neurodevelopmental syndrome and increased expression of the CHD2 gene in cysts. And then they said, treasure your exceptions. They suggest that link RNA genes upstream of known Mendelian genes may be a rich source of variation-producing Mendelian syndromes that we haven't tapped because we haven't looked carefully, we didn't use whole genome sequencing and so forth. So whether they're right or not, I don't know, but I think it's a very good idea.
I have a question. So,
Yeah.
can I see the deletion a couple slides before?
Okay, go ahead.
So for like patient 1, it looks like the, I guess, beginning part of the ORF for CHD2 is deleted.
I think that's just an SMD out there.
Oh, I see. It's like zooming. Okay.
I'm sorry.
Okay. Sorry,
Okay.
I was reading the map. That's what I was
Okay.
confused.
All right, let's keep going. So now then the last thing we'll connect to complex traits. To what extent do Mendelian disorders contribute to complex traits? You know that complex traits are those disorders that generally result from variation in a number of genes that are sort of additive to produce the phenotype, and plus a strong environmental influence. So it could be that Mendelian disorders contribute. So it's sort of the gene burden model, disease genotype in the primary locus with milder variants, aggravated locus, loci, and that gives you a phenotype we call a complex trait. Alternatively, you could be homozygous at the disease gene locus and heterozygous for variants in related genes, increasing, determining the severity of the phenotype. And then the other place to look are, if you have a phenotype that you typically think is due to a complex trait and there's a distribution of severity, you might look at the tails of the distribution, the severe end and the mild end, and see if those are really Mendelian traits lumped in because of phenotypic similarity to complex traits. So those are some ideas about how Mendelian disorders, and I'll show you an example of that in a moment. Next slide. Oh, so now I got to switch. So everybody can stand up and stretch.
I wanted to mention the fact that we will not then have a discussion of incarnates.
That's what she said.
To reiterate what Ms. Farland was saying for the last second, the link RNAs, we're not going to cover too much, at least right now, in the class, so don't worry too much about those. Focus more on to Mendelian disorders, the severity of the actual expressivity, and everything else that was covered in the lecture.
So what have I got to do here?
We broke every single one of them. She used to be obsessed with it. She used to decorate her house with them. I never had a glass house until years ago. And then Irvin's mom gave me a Mexican code. There's only like 6 ingredients in it.
He has like 25 articles and stuff.
Athg has?
That all cite that one. So,
Okay,
I don't know. There's a lot to wait.
so my reference is out of date then, so we ought to look at that.
Recording in progress.
So if I want to, now I want to share my screen. Here.
What could be happening is that you need to go start the presentation.
I don't know, I can't tell. I think it's this one.
You open the PowerPoint first, and then you can share the screen.
Yeah, that's the plan.
I got cut off before. I wanted to mention that the lecture about the cabinet will be presented by Loyal, the Jack course, and so it's not our course for this year.
Oh, it does that resolution thing?
Yeah.
That's not PowerPoint. You would say that. You only need that.
Minor physics, so just take a second, we'll be shortly.
And also you have RNA sequencing.
Yeah.
That is? Okay, now we can.
I think they are isoforms because it's from RNA sequencing.
How many papers?
There's 25 on PubMed.
So, Dan is saying that Athg has 25
I
papers.
searched cited by, like, cite this paper.
Yeah.
The original, and then here's the author.
So he's basically
Oh, that's
a computational man.
You met one.
So there's all sorts in here.
All right, well, I'll report back.
So I don't know if any of them actually touch on questions.
Desktop, there was a picture titled Black Scientists. And I was just sitting on the desktop. Yeah.
Maybe she's doing it in Black History Month.
Yes, I hope. Weird.
I can see it online.
And she had some involvement in the Black community and it just had
Yeah.
like random folders.
Yeah, you can see it
Black
online.
Scientists. She had one of her bookmarks was Tito's. Just said Tito's.
Like the liquor.
Yeah.
Perfect.
Everyone
So
has
you
a
see
vice.
the challenges and opportunities.
See that? So Elena, if you didn't hear, she mentioned that there's no lecture in this course on link RNAs, but we can go to Bar Harbor this coming summer Loyal Goff will give a lecture on linked RNAs. So stay tuned, but you can also read that paper on Chasser, and I gave you the reference for a very recent review on linked RNAs. It's pretty good.
And also writes really great papers.
Pardon?
The senior author on that paper, the Chaser paper, writes great papers, in my opinion.
Ann O'Donnell.
Yeah.
Yeah, she'll be speaking. Actually, she'll speak at Harvard as well. Not all her papers. She's very good, but not all her papers are great.
Oh.
That one I critiqued last time. So the paper.
The ones I've read are great.
Okay. All right. So now we're going to talk about complex traits. And it's 11:30. Okay. Next. So sometimes people refer to these, particularly in the past, as multiple gene traits. So they're phenotypes determined by combined influences of variants of multiple genes plus environmental factors. So, and they include lots of common disorders: diabetes, hypertension, neuropsychiatric disease, coronary artery disease, and many, many more. Okay. So remember I said if you go to the hospital and you score how many people are in the pediatric hospital for reasons of Mendelian disorders, you get about 10%. If you go over there and score for complex traits, you'll get about 60%, something like that. So in contrast to Mendelian traits, complex traits involve variants that mildly perturb the function of protein products. So any one of those is not sufficient to cause the phenotype. So these are risk alleles or susceptibility alleles. They're not causative alleles. Okay, next. So genetics has been struggling with complex traits, particularly as regarding medicine, for quite some time. So we want to identify the variants in the genes. We want to identify and avoid the environmental variables that contribute to the phenotypes. We want to make the diagnoses pre-symptomatically, and then focus on prevention. And keep people healthy instead of rescue people that are sick. So I used to say, well, maybe soon in the future there'll be a unit of the hospital instead of the emergency room will be called the prevention room. And you'll go to the prevention room to get your prevention for something you know you're at risk for. Obviously, that should be your personal physician that does that. Okay, and a classic example is type 2 diabetes. So its incidence is increasing, and it's entwined with increased incidence of obesity. If you have a sibling who has type 2 diabetes, your risk is about 3.5 times higher than the general population. And since the general population risk is pretty high, 3.5 times pretty high is really high. Hard.
Is that like controlled for being in the same environment as your sibling?
Yeah. In twin studies. Typically not, but in twin, people have tried to get at that by doing twin studies. And type 2 diabetes is a chronic illness with an array of complications. I always like to do this experiment. How many in the room have a relative with type 2 diabetes? Seven. So about half of us. It's pretty common. And most of the complications are, a lot of the complications are microvascular, that is to say defects in blood vessels, presumably caused by toxic effects over time of elevated glucose on blood vessels. So they can be microvascular. That's the fundus of an eye. This is the optic nerve. And these are all diabetic changes in the retina. So that one complication of type 2 diabetes is retinal vascular disease and blindness. Next slide. Another is peripheral nervous disease, because the vessels to the peripheral nerves are damaged, and then the peripheral nerves die off. Does anybody know what this is?
Is that a heel of your foot?
That is a heel. And this person has peripheral neuropathy, so it doesn't have sensation in the skin over his heel. So he's actually, that's his heel bone you're looking at. There's no skin there. Next slide. Does anybody know what this is? This is a coronary artery. And what do you notice about that coronary artery?
That big black spot.
It's got a clot in it. So that's a myocardial infarction. That's post-myocardial infarction. So lots of vascular diseases, vascular complications of type 2 diabetes. So it's a really heavy burden of disease, both in terms of the numbers of individuals and their complications and the demand on the medical system. And if you go to other parts of the world, it's even higher. For example, in the in the Mexican population, over 50, individuals over age 50, the incidence of type 2 diabetes is something like 60 or 70%. A huge demand on a huge burden on the individuals and on the medical system and on the economics of the country. Next slide. We're going to come back to type 2 diabetes a little bit. Before we do that, let's just think about the biology of disease. So you've heard me say this before. In my view, all disease has a genetic component. All disease has a genetic component, formerly referred to as heritability. So we all, by shorthand, say this is a Mendelian disease or this is a genetic disease. So I say this is an overtly genetic disease. That's Mendelian disease. But I know that every disease has a genetic component to it, just as every disease has an environmental component to it as well. All disorders are complex. So we call this sort of multifactorial disease complex traits. Even Mendelian disease are complex. So everything's complex. What we do call complex traits are quite common. We've already discussed that. And they tend to occur later in life than Mendelian disorders. Just remember I said for Mendelian disorders, most of them present at the severe end of the spectrum, and that's in the pediatric age range. Most complex traits, particularly those that involve physiologic homeostasis, present in adult life. The one set of complex traits that present in the pediatric age range are malformations, and that's because they're involved in development, which is the early phenomenon. Next slide. So I sort of, although people separate complex traits and Mendelian disorders, and you find whole groups of geneticists that focus on complex traits and whole groups that focus on Mendelian traits, I see them as part of one spectrum. It's interesting to point as an aside, when did the sort of bifurcation of complex traits and Mendelian disorders begin? You guys know this. Daniel?
Kind of from the beginning, Mendel and the Ancestrian debate.
You're exactly right. So it's the Mendelian Ancestrian debate from 1900 to 1920. Mendel was sort of the hero of the Mendelists, and Galton was the hero of the complex trait people. It's interesting. They were both born in 1822. And so their lives were intertwined intermittently forever thereafter. And we continue to deal with this separation. But I think of it as the phenotype comes from some sort of physiologic or homeostatic imbalance. And you have, in the Mendel case, one variant gene has a big impact, but it's influenced by modifier genes and influenced by environmental variables. I can take a child with PKU and perturb their environment severely by putting them on a special diet, and they'll develop normally. So even under that really severe disruption of physiologic homeostasis, I can control the environment to modify the consequences. And on the other side, you have many genes that contribute each to a lesser extent, and environmental variables contributing to a greater extent. Okay, next. We've seen this before. The risk variance here, next. Next. The risk variance for complex traits are down here, variance of modest effect. So you don't really see a phenotype if you just have one. You have to have sort of a combination of several to and a certain level of environmental stress that puts you over the edge. Next slide. So let's just talk about the genetic component, heritability next. So heritability is actually a classic genetic feature, and it's a fraction of the phenotypic variance explained by genetic factors. And you can estimate it from the correlation of measurements of the phenotype in individuals of known degree of relatedness, for example, SIDS. And it's in the classic genetic literature, it's referred to as H2, and it ranges from 0 to 1. And H2 high level is strong, higher, the stronger the genetic contribution. So H2 for stature is 0.8, or about 80% of the variance that contributes to stature is due to genetic variation. For body mass, it's similarly high, 0.7. Okay? Next slide. Now, why are complex traits common? Next slide. So variance of modest effect, less subject to natural selection. Selection can't actually get a handle on it because it has a very modest effect on the phenotype. And therefore, they're tolerated at higher frequency in the population. Variance in many genes and biological systems confer risk for the same disease as identified by physicians. This is the heterogeneity of etiology. So if you have lots of different genes that have mild variance that contribute to a phenotype, then we may give them two individuals the same diagnosis, but they don't quite have the same genetic contribution, and they may not quite have the same environmental contribution. It depends if you're a lumper or splitter. And many environmental variables are common. And I may have mentioned this to this group before, but I can still remember driving by a McDonald's restaurant where the sign was over 50,000 hamburgers sold. And now, what is it? I don't know, 500 billion hamburgers sold, or something like that. The total caloric input in the American population from the year 1990 to 2000 went up by something like 13%. So the genome is always, the environment changes more rapidly than the genome. The genome is always trying to catch up to the environment. And the way it catches up is by natural selection, and particularly against those that have unfavorable genotypes. They have to see their doctor. Next. So goals for genetic medicine: identify genetic and environmental variables that contribute risk, predict risk for each patient, and individualize risk, understand the pathophysiologic mechanisms responsible for the phenotype, and use this knowledge to improve prevention and treatment for each individual patient. We'd like to keep people healthy, but when that fails, then we'd like to rescue them by developing some kind of treatment that restores them back to health. That's a common, that understanding of the role of medicine is quite common in pediatrics. Pediatrics is where you vaccinate, you give kids immunizations, and you do things to keep the kid healthy. In adult medicine, it's not as common. That idea is not as common because people that come to see their doctor in adult medicine usually have a problem. Next slide. Now, one of the challenges of complex traits is this heterogeneity of etiology. We already talked a good bit about heterogeneity, but here's another aspect of heterogeneity. So imagine that you have a particular disease, and medicine, physicians make that diagnosis. It could be type 2 diabetes, or it could be schizophrenia, or whatever. And let's imagine I had a huge population of those individuals, and I call them all the same diagnosis. If I could really know the cause of that phenotype in each individual in that group of patients, what would I find? So I might find a few individuals that have a rare Mendelian cause for that phenotype. They're just lumped in there. I might find a few individuals that have a rare environmental cause for that phenotype. And I find a bunch of individuals that have an incompletely overlapping set of genetic and environmental risk factors that just combine in unfavorable ways and they have the disease. Okay? For example, next slide. Let's talk about schizophrenia. Next slide. Patients with a rare inborn error of metabolism called homocystinuria, defect in methionine metabolism, have psychotic crises which are virtually the same as the psychotic crises in schizophrenia. And I had a patient, I sort of not thinking about the psychotic crises, came to see me in the emergency room, did some stuff, said, "You're fine, go home." Came back the next day, I said, "I just saw you yesterday, you're fine." Did some more stuff, "You're fine, go home." Came back the third day. And I said, "Everything's okay." And he said, "Yes, but what about this telephone pole that's sticking in the side of my chest?" What? So he then spent 6 weeks on the psychiatric ward trying to get control of this psychotic break, basically. Next slide. PCP injections, street drug, cause a psychotic phenotype exactly like schizophrenia. So that's an environmental one. Next. And then we now know that variation in over 300 genes and a lot of environmental factors contribute to schizophrenia. So I take this collection of patients, they all have the same diagnosis, but if I could really understand why they're sick, why each individual is sick, it would be different. And if it's different etiology, then maybe I should be treating them differently. I could treat the homocystinuria with a methionine-restricted diet. I could treat the PCP patients by getting PCP off the street. And I could treat the others, maybe this individual has risk variance of 150 of those 300 genes, and the next one has risk variance of a different 150 of those 300 genes, and I should treat them differently. So ultimately, I think we're going to know all this, but we don't know it now. That's for sure. Next slide. So carried to the extreme, each person has their own disease. So this is hard for it's hard for medicine to scale to that extent, right? That's why we can't do that. So what we do is we give this diagnosis, then we sort of pick away at it. We find a few people that have this reason and a few people that have this reason. But we don't really have a global picture yet. I would argue that that's what computers are for. They can keep track of all this stuff. We get smart enough to find all the variables, put them in a computer, and the computer can keep track of it and tell us what the problem is in this individual versus that individual. Both with the same disease, same diagnosis. Next slide. So what's the progress in disease gene identification? These data are loosey-goosey, but because it's hard to keep track of. But you can see that back in the 80s, we knew almost nothing. And in the late 80s, around 1990, or well, no, the 1909 was the genomic. Anyway, the Mendelian disease gene has been going up, and we're now at about almost 5,000. We didn't do very good with complex traits until GWAS was invented. And that was around in the 2000, 2005. It's now started going up quite abruptly. So there's a huge amount of GWAS data and a huge number of genes for which variants have been shown to contribute some small risk for a particular phenotype. Okay. So how do we find it? So one would be candidate gene approaches. So you can say, well, the patients with this disease, this complex trait, the abnormalities that they have suggest that a certain system is not working well. I'll look at the genes that are encode proteins that are in that system. That's the candidate gene approach. So survey all known components of the biological system. It's quick because you have a limited search space, and less expensive because you're not looking at so many genes. Many prove its successes, but it suffers from looking under the lamppost. In other words, we only look where we think there's a problem. So we don't find out anything that new. So this method still occasionally works well, but it's not what's really responsible for that big increase in that slope of that curve I showed you. Next slide. So a different approach would be what I call whole genome agnostic approaches. By agnostic, I mean the only idea I have is that somewhere in the genome there are variants that contribute to this disease. I don't make any assumptions about what biological systems, what genes, anything. I just make the assumption that there is genetic variation that contributes to the phenotype. So it's generally more expensive and time-consuming. It's agnostic as to cause and often identifies components or biological systems that we didn't even know contributed to this phenotype. So it's a big bang for the buck. There are many successes. Next slide. So one way to do this is linkage. And to do linkage, we already talked about, you ideally good pedigrees, score patients for affected, unaffected, and you're basically taking advantage of Mendel's law and Mendel's laws in human families. Next slide. So you have some panel of genetic markers that cover the genome in a fairly uniform way, and then you have the segregation of the phenotype and the pedigree. So here there's, we're just looking at 2 markers, and the markers are fully informative. By that I mean you can see both alleles in all the individuals. So for marker 1, the top person's genotype is AB, and for marker 2 is CD, and for the other parent CD, and then for marker 2 WX and 1Z. So in other words, you can see all 4 of those. You can follow those 2 genes, all 4 alleles in this pedigree, and then you just see what's passed on, and you look for co-segregation with the phenotype. So the affected individual transmits her A allele for marker 1, and this person gets the C allele from the father, has the disease. He transmits, again, the A allele and the phenotype. So that's a little suspicious that some gene, doesn't mean the marker A is responsible, it just means that some gene in the vicinity of the genome is contributing to the phenotype. Okay, so you can see if you do that in a much more expansive and granular way, you can begin to make statistically significant assumptions about the linkage of the marker to the gene of interest. Okay? So you're searching for shared chromosomal segments as defined by those genetic markers. For families, you can combine families, but you have to be aware of locus heterogeneity. So if you have locus heterogeneity and the phenotype is due to in one family is due to mutations in one gene, and in another family mutations in a different gene, then that screws up your linkage, right? Because you're comparing apples to oranges. So you can combine families, but you have to be aware of locus heterogeneity. It's not affected by allelic heterogeneity. It doesn't care. This is a method that tries to find the location of the gene. It doesn't care about what the allele is in the gene. That will come later once you find the gene. And there's a statistical parameters that we use to define success, and it's typically a log of the odds, or so-called log score, of 3 or greater. 1,000 times more likely to be linked than not. That's the log score. Okay. Does that all sound familiar? Problem is, you got to find these big families. The genetic part of it is easy, but finding the big families and phenotyping them takes time. So it's slow. And the other problem is limited resolution. When you finally get a log score of 3, you say, okay, whatever the gene is, it's in this region. That region might be 5 or 10 megabases and might involve 100 genes. And I gotta go sequence those genes and try to make sense out of it. Okay.
I have a question.
Yeah, please.
There's a paper that you and Nara worked on where you did the metacontraventus.
Metacontraventus, yeah.
So how do you explain like a log score? Like I think one of the loci in there had a log score of like 1.8, and there were other log scores that were higher, but
Yeah. So this is the log score you have to say to find the locus.
I see.
Okay. Well, we use the log score in the reverse way. We said whatever these log scores that we can measure in this family are not statistically significance. However, every place that has a negative or no log score at all must not have the variance. So we threw it away.
Okay.
So we used it just to clean out. We narrow the search spaces. Yes, Carter?
So is this logged or is it adjusted depending on the number of markers that you're using? Is like if you were to expand the number of markers, do you need to increase the log odds that
Yes.
you're using?
So you try to, when people calculated the number of markers that would be required, they said, okay, I want to uniformly as possible, cover the genome. And what, of course, limits how well you cover it is the frequency of recombination. In other words, if you have a marker on this part of the chromosome and a marker on this, they're farther apart, the likelihood of recombination is at least 50%, and therefore they're non-informative. So you have to have a network of markers that is sufficiently dense that you can compensate for recombination. Typically in linkage calculations, you use a parameter called theta that says that at a recombination frequency of X, I can find this. So that's what determines the necessary density of the markers. And we used to use short tandem repeats, which are not as dense, not as many and not as dense across the genome, but now modern linkage is done using single nucleotide polymorphisms in combinations, like 3 close by, stuff like that. But when the Center for Inherited Disease Research, the community runs, got started, this is what we were doing. Okay, next slide. So here's another method, association. And this is the ancestor to genome-wide association studies. Okay. So the way this is, okay, we will take a population of affected individuals and a population of controls. And we'll have a dense set of markers. And we'll look for those markers that are enriched in the affected versus the controls. Now you have to be careful about what you call affected and what you call controls. Okay? Pretty simple idea, really. However, when this idea was first really proposed, we did not have such a dense set of markers. Why is that? Because we were just learning how to identify and genotype single nucleotide variants. But now we got a ton of them. Okay, so a typical chip that's used to do genotyping for a GWAS study uses on the order of 1.5 to 2 million single nucleotide variants spread out over the genome. When we first did this using single nucleotide variants, the chips had maybe 300,000 markers. Next slide. Now this, in my view, is the key paper, and I'm going to ask you to read this by Neil Weish and Kathleen Maripanius in Science in 1996. And they argued on a theoretical basis that association would be much better than linkage. You wouldn't need big families. You could do it quickly, and you would get higher resolution. You just needed a dense set of markers. So they said these single nucleotide variants that we're just realizing are so common would just be the right thing. And they did a whole bunch of calculations. So this is a very important paper in my view. It was the foundational paper for all genome-wide association studies. Okay, so you're searching for shared linkage disequilibrium haplotype blocks in the genome, and we'll talk about that in a second. And each block is identified by SNPs that map into that, by single nucleotide variants that map into that block. Okay. And those are the ones you genotype, and they're called tagging SNPs. They tag that block. It's affected by allelic heterogeneity because the linkage disequilibrium blocks are, on average, maybe 8 to 10 to 12 kilobases long. So a gene might be 100 kilobases long. So a gene might have 10 linkage disequilibrium blocks. So in this person with this disease, the mutation may be in block 2. In this person with the disease, the mutation may be in block 6. So allelic heterogeneity isn't bugaboo here, but generally people have enough individuals so that you can count it has higher sensitivity, so it detects variants with odds ratios of 1.1 to higher. Remember I said for linkage you had to have an odds ratio of 2. Resolution, instead of finding 5 to 10 megabases, it finds 10 to hundreds of kilobase, much smaller hit size. And you need a dense marker set, which we have a dense marker set. Now, next slide. Okay, now I'm going to talk just as an aside here about linkage disequilibrium. You guys have all heard of linkage disequilibrium or not? Maya's shaking her head yes. Other folks have heard something about it. So this is the way I view linkage disequilibrium. Okay, so this is a little chunk of the genome. And each of those aqua blocks are linkage disequilibrium blocks. And they are separated by recombinational hotspots. Okay? So you have 1, 2, 3, 4, 5 LD blocks across this stretch of the genome. And if you could sequence each of these blocks, you would find single nucleotide variants that were located in the block. And you could construct some haplotypes of the various variants in each block. And it turns out if you use relatively frequent single nucleotide variants, there's typically on the order of 3 to 5 haplotypes that cover most flavors of a particular linkage disequilibrium block. Okay, so here I show 3 SNP haplotypes that cover this block. And if I genotype these SNPs that define the haplotype, then I can tell which sort of flavor of this LD block this person has. Okay? And these are called, you can think of these again as tagging SNPs. They tag that block. So they'd be on my chip, my genotyping chip. And the same is true across all. And so here's a plot of linkage disequilibrium high in this block and then low across the recombination hotspot and so forth. So, okay? Everybody got it? Next slide. Okay, so let's just look at one LD block. And this block is characterized by 9 SNPs, or 9 single nucleotide variants. They're located at a particular spot in the LD block. Next slide. Next. So here's the haplotype of a common set of genotypes in this LD block. So here's one haplotype. Now, all of a sudden, a variant occurs, a mutation occurs. Now, when the mutation occurs, it happens on one particular haplotype, right? So if I call these, I want to tell, I have this, if I'm looking at this haplotype block, I could just say, well, I'll measure SNP 3 and SNP 7. And those would be my tagging SNPs. Next slide. Now, recombination over generations sort of chews away at the LD block a little bit. Next slide. Until I sort of have a core LD block. And it's, I still have the tagging SNPs here. And here's my variant that I'm trying to learn about. So that's my disease-associated haplotype. Okay? Now, of course, if I measure the haplotype and it's something different, then I'm not going to, I'm going to say this one is associated and this one is not. Right? Okay, so that's the non-disease associated haplotype. So the only problem is, of course, when the mutation occurred, there are other people in the population that had the same haplotype. So not everyone with this haplotype has the mutation. It's just enriched for people in that haplotype. So I have some people that have the same haplotype, no risk variant. All the people with the risk variant have this haplotype. And then I had the non-disease risk haplotype. Those are my possibilities. Okay? You'll get all these slides. You don't have to. So now I say, okay, I'm going to try to do a genome-wide association study, and what am I going to learn? Next slide. So I'm going to find that this haplotype, this ACE haplotype, is enriched in those with the disease because they're tagging this LD block that's carrying this disease risk variant. The tagging SNPs are not the variant. They just tag that block that has the variant. But I know I've already said that other people in the population are going to have the same haplotype, but not the risk variant, because when the risk variant occurs, it's only happening to one person. But nevertheless, that will be enriched. That haplotype will be enriched in those that have the disease, right? Next. Okay, and then I'll find other people who have a different haplotype, won't have the disease risk variant, and that will be the non-disease haplotype. So, one problem is heterogeneity of etiology, right? So if I lump everybody together, and let's say I lump 1,000 people together, and some of them have the disease because they have that risk variant, and some of them have it for other reasons, I'm going to find some people that are homozygous for the non-disease associated haplotype that have the disease. That's going to be a screw up my analysis to some extent. Next slide. Go back. So, but if I have big numbers and good phenotyping, I'm going to find enrichment for that disease-associated allele. Now, so then that identifies this LV block. Average size of LV blocks is what in the genome? Guess. Depends on your ancestry. So if you have African ancestry, the average LV block is about 8 to 10 kb. If you have Northern European ancestry, because of that population bottleneck, the average LV block is about 20 kb. There are wide variation of some LV blocks are hundreds of kb. Okay. Next slide. So I go through all of this, and I say, in my disease population, here is a set of tagging SNPs that tag a particular LV block that is enriched in my disease population as compared to my controls. Okay? So what could be going on? Well, the tagging SNP, you could be really lucky. The tagging SNP might be the causative variant. Much more likely, however, that the tagging SNP is in the same LV block. It's just not the causative variant, right? So I have to decide, once I get down to an LV block, I have to decide if the variants I'm using to find that are the actual causative variant, that's a direct association, or whether they are tagging SNPs and they tag the LV block that contains the causative variant. Okay? So that's indirect association. So if I get positive association, I say the marker is a causative variant, or the marker is an LV with a causative variant. Or it could be an error. This population stratification, design of the experiment is wrong, or something like that, be false positive. That means I need probably to replicate the study to eliminate, to reduce the likelihood of false positives. If I find no association, and this is a pain in the butt about association studies, is that a true negative? I can forget about that part of the genome? Or is it the power is the study is too small and it's underpowered? There is actually a gene there that has variants that contribute to the risk. I just need to have a bigger study. You know, I just need to go spend another $10 million to recruit another 500,000 people. So that's a frustrating aspect, at least I think, for GWAS studies. Okay, is everybody with me up to that point? Okay, next slide. So, is it worth it? Well, absolutely, it is worth it. It's the biological significance of true positives. First of all, you got there with an agnostic approach, so your preconceptions of how this disease works didn't contribute to this. So you're going to find new stuff. And the agnostic approach identifies the biological system involved in the phenotype. Second, and I'll just say a word, with the first GWAS study that was done, and I don't have that slide in here, is a study for age-related macular degeneration. It's a tiny little study. There were 100 affected individuals. Now we do 10,000. And it got two statistically significant hits. One turned out to be a false positive, and the other turned out to be a missense variant in a gene called complement factor H. So no one had any idea that innate immunity, complement factor gene, had anything to do with macular degeneration. So we got there agnostically, and wow, this exposes some new biology. Now it happens, once someone found that and looked back at the literature, actually there were clues there that the complement factor H was important, but no one was paying attention to it. But this, all of a sudden, everything made sense. You know, the light bulb went off. So that's a real power of this approach, I think. Subsequent identification of causative variants. Then you want to find the causative variant, because that'll really tell you what's going on. And understanding the pathophysiology then increases the opportunity for prevention and control. Next slide. So here's how the results are portrayed. You've all seen these so-called Manhattan plots. This is the chromosomal on the x-axis, and a measure of the statistical significance of the association of the genotype at a particular SNP to the phenotype. So every one of these dots is a particular single nucleotide variant that was on the genotyping chip. And down here, where there's not much association with the phenotype, it's just a glom of dots, right? So in order to avoid going after things that were really low risk or had a high chance of being false positive, the statistical bar for success was set at a fairly rigorous level, okay? Because you have a Bonferroni issue here, right? You've done a million tests, so you have to, it's a high correction for that. So next slide. So the bar for sort of success was set at a high level, 5 times 10 to the minus 8 across the genome. So now you can see, I have this initial study for type 2 diabetes. I have one set of variants in a gene called TCF7L2 that are well above, that are very highly associated with type 2 diabetes. I have another one that's right at the margin. And then I have a whole bunch, like things like this. Is this just chance, or is this really an area where there's a real association? If I double the population size, maybe I'd get it up over that. I don't know. I have to do the experiment. Okay, well, does anyone know what TCF7L2 does? It's a transcription factor that controls the expression of insulin. So you can imagine doing this association study for type 2 diabetes and saying, I don't know what I'm going to find. And then you find this big statistically significant association, and it's a gene whose biology is perfect, right? And that's held up. That's the strongest variant to contribute risk for type 2 diabetes. Next slide. So some common themes. Now we've done a huge number of GWAS studies. If you need to detect a small risk, you have to have bigger samples. More money. Replication necessary to confirm, because you want to make sure that something about the design of the experiment, the way you decided who was infected and who wasn't infected, didn't bring some bias into the study that you're touching. It exposes unexpected components of the disease network. That's the real power of it. Most risk variants for complex traits are in regulatory rather than coding space, whereas most Mendelian are in the coding space. Okay. And that's, they tag LD blocks that are outside the coding space of a gene. Okay. And of course, understanding gene regulation of gene expression is harder than understanding coding changes, so that's another challenge. And we find unexpected overlaps between genes contributing risk for different diseases. For example, psoriasis and diabetes share some overlaps. We thought medicine said it was 2 different diseases, but in fact the genetic underpinnings overlap. So So a lot of interesting biology. Next slide. So this is about regulatory motifs. I think it's self-explanatory. A lot of new methods to figure out, and Mike Beers after the Sotomayor. Next slide. And you can go to a map. That NHGRI had a map of all of the results of GWAS studies. So here's the karyotype. And each of these lollipops is a GWAS, a statistically significant GWAS hit for different systems. So neurologic disorders are yellow. And they stopped keeping track of this a few years ago, but it's too just blur now. So it's huge now. But you can go there, go to this website and see the latest, greatest.
Coming up on 12.
Yeah. Next slide. Next slide. This shows the current state of the art. So it's publications in blue, studies in red, the number of studies, what kind of studies we're talking about, top associations, so forth and so on. This is a wonderful article on the history of GWAS, studied by Peter Vischer's group. I urge you to read it. It's American Journal of Human Genetics in 2017. Next slide. The diversity problem. It turns out that you'll find very different variants that contribute to risk for a particular phenotype in one population that are not exactly the same as in another population. And if most of the GWAS studies are done in individuals of Northern Europeans, then you find out something about Northern European ancestry, but it's not 100% translatable to other populations. So we have to do more GWAS studies in other populations. And that problem is being addressed to some extent, but not as robustly as we would like. Next slide. And the success has been good for some disorders like coronary artery disease. I think it's pretty darn good for inflammatory bowel disease. Schizophrenia, we have a long, long way to go. Next slide. Most of the hits in schizophrenia seem to be in proteins that are involved in synaptic biology. Next slide. This, I think, is really promising. This is looking at a huge number of GWASs done for type 2 diabetes. Remember I said they have all different kinds of symptoms. Now people are being able to subdivide people who come with a diagnosis of type 2 diabetes and determine at least 5, sometimes depending on how they do it, as many as 8 subgroups that have different risks for the different complications of type 2 diabetes. Individuals who have large vessel disease, these are the individuals you see going around the neighborhood in wheelchairs that have an amputation. That amputation is almost always due to diabetes, and that's a large vessel complication. And there's a set of people with type 2 diabetes that just have malignant vessel disease, and they end up having to get amputations. There are some others that have retinal degeneration, others that have coronary artery disease. So now you can take the population, and depending on their genotype at lots of different locations, you say this person has type 2 diabetes, but they are at special risk for large vessel complications. Or this person has type 2 diabetes, they're at special risk, especially high risk for retinal degeneration. And you can begin to treat them accordingly, according to their individualized risks. Next slide. We're not going to have time to cover polygenic risk score. Let me just say very briefly. So these are our statistically significant hits, right? So what about this? Is this a real thing? Well, it looks sort of real to me because there's a lot of associations in the same place that just don't get to the significance. So maybe that's just a weaker signal, but it's true. So that suggests, well, maybe there's more information down here below the 5 times 10 to the minus 8th level that actually contributed to phenotype. And is there some way to harvest all of this sort of weakly significant information? The argument is further made that the false positives tend to occur at random, and the weakly significant association tends to recur over and over again. So if I can figure that out and make calculations, I can get more information out of this data set and you should read, this is a great paper. Okay, here, next slide. So what if I draw the level lower, and then I sort of look through all of these sort of intermediate value associations. Next slide. So this is how you calculate a PRS. You do a GWAS. You put it into a computer and follow a bunch of software and cranks out a score. The score is normally distributed, that is to say it has a normal smooth curve of risk versus number of individuals in a population. You typically validate it by looking at independent cohorts, and then you develop a curve that gives a percentile of risk from very low to very high, and then can use that clinically to counsel individuals about their risk. Next slide. It's normally distributed, and these people at the top end of the score have a very high risk, almost as high as Mendelian risk. Next slide. Next. And these data really impressed me. So this is normal. They start off looking at individuals for which they had newborn data, the birth weight. Okay, and they did a polygenic risk score for weight, and they divided it into deciles. So this is the lowest decile. This is the highest decile for risk of weight. And there's no difference in statistically significant difference in weight when in the newborn period by their PRS. But if you follow the same individuals over as they get older, at 8 months, here it is at 18 months, here it is at 3.5 years, here it is at 8 years, and here it is at 18 years. And the difference between these two deciles at 18 years is about 13 kilos. So you calculate a polygenic risk score and say people in this decile are at risk for being bigger. So the risk for severe obesity is about 25-fold greater in the top decile as
Recording
the
stopped.
lower decile. So there's some biology there, real biology. Okay. Now you can use that medically. Next slide, and we'll stop. You can read this next slide. This is back. Go back. This just shows that just like GWAS studies, the calculation of PRS on GWAS studies from different ancestries doesn't work very well. So this is a famous paper now by Martin in Nature Genetics in 2019, looking at the prediction accuracy versus population ancestry, and the original GWAS was done in the Europeans. It doesn't work very well for individuals from other populations, so you have to do it in other populations. Next, and I'm going to put one more. Next. So here, can you combine this with Mendelian disease? So this is a PRS for glaucoma. Glaucoma is an insidious onset, increased interocular pressure, major cause of blindness. In some families, it's almost Mendelian, and it's due to a variant in a myosin C variant, but that accounts for only a small number of patients. So here's what happened if you combine the PRS score plus the genotype at the myosin C, and you can get a huge increase in risk versus the defined risk population. And so you can measure prospectively intraocular pressure in those individuals, treat them in an earlier way, and hopefully prevent the deleterious consequences of increased intracranial pressure, increased interocular pressure. So that's, in this case, combining some Mendelian information with some PRS information. The uptake in the clinics has been very slow, because the risks are not great. It's complicated. You have to have genotype data. You have to have a reference genotype data, GWAS data from populations ancestry matches the person you're trying to counsel. But it is a way to get at risk that could be useful in certain situations. I think this glaucoma one is a great one. Medicine changes very slowly. If you think, do you guys know what the Exxon Valdez is? There it is. Exxon Valdez was a big oil freighter that floundered up in Alaska, and oiled the beaches of Alaska. And they could see they were running aground, but to turn one of those tankers takes forever. So they knew they were going to hit the ground, they couldn't turn it. So medicine is the same. It's very hard to get medicine to change. But it's going on. Next slide, I think that's the end. Keep going. All right, sorry to run over. Thank you very much.
You want them to read the?
I'm going to send you the papers.
I downloaded the one.
You should read them.
Yes.
Yeah, I want that one. And I was going to send you the reference for the. Well, you're welcome. I'm sorry, what? Yeah, I'm just saying. Please.